Hey everyone, in today's video we're going to walk through the transduction pathways. So epinephrine, a hormone, binds to the specific receptor on the cell membrane known as the G protein coupled receptor, or GPCR. So when epinephrine binds, it changes the shape of the receptor, which in turn activates the G protein inside of the cell. This G protein has three parts: alpha, beta, and gamma. The alpha subunit breaks away and moves along the membrane. The alpha subunit binds to an enzyme called adenylyl cyclase, which is also embedded in the cell membrane. Adenylyl cyclase then converts ATP to cyclic AMP, or CAMP, which is our second messenger in this pathway. CAMP activates protein kinase A, or pKa, by binding to its regulatory subunits, causing the catalytic subunits to become active. pKa then phosphorylates other proteins in the cell, leading to a variety of responses. One important response is the breaking down of glycogen into glucose, which the cell can use for energy. So that's the epinephrine pathway. To summarize, epinephrine activates GPCR, which leads to the production of CAMP, activating pKa and ultimately causing a cellular response. So next, we'll go over the MAPK pathway, which plays a major role in cell growth and division. The pathway begins when a growth factor binds to the receptor tyrosine kinase, or RTK, on the cell surface. This binding causes the receptor to dimerize, or pair up, and undergo phosphorylation, which activates the receptor. The phosphorylated RTK recruits and activates an adapter protein that in turn activates RAS, a small G protein. When RAS is activated, it initiates a cascade of kinases. It activates three kinases: RAF, then MEK, and finally ERK. Each kinase phosphorylates and activates the next in the series. Activated ERK enters the nucleus and activates transcription factors that leads to changes in gene expressions, promoting cell growth and division. To recap, the MAPK pathway involves a receptor tyrosine kinase, activation of RAS, and a kinase cascade that ultimately affects gene expression in the nucleus. Now, let's talk more about these second messenger pathways. In the GERK pathway, a GPCR activates a G protein which interacts with a potassium channel called the GERK channel. When activated, this channel opens and allows potassium ions to flow out of the cell. PLC splits the membrane phospholipid into two second messengers, IP3 and DAG. IP3 moves into the cytoplasm and binds to the calcium channels of the ER, causing a Ca2+ release. The estrogen pathway is a bit different because estrogen is a steroid hormone that can cross the cell membrane. It binds to its receptors inside the cell, forming a hormone receptor complex. The complex moves into the nucleus where it binds to the specific DNA sequences, acting as a transcription factor to regulate gene expression directly. We explored three pathways, each with unique mechanisms for signaling within the cell.